Hey guys, so welcome to Microbiology. As you are all aware, we have moved to an online version of this course. Um, I just want to mention a few things as this is the first lecture. Um, Hopefully, you all have either obtained or will be obtaining a textbook. Um, the textbook is Tutora. We are currently on the 13th edition. Um, you can either get an ebook or a hard book, and I'm sure if you even look really hard on eBay, you might even be able to find a PDF version of it. I know that there are a few of those floating around, um, but I highly do recommend that you have a textbook that you are able to refer to, especially seeing that this is an online course and we are not face to face. I would also like to mention that you do not need mastering. So that should save some money without having to purchase mastering access. Um, and like I said, you can just get the textbook. If you get your hands on a 12th or 11th edition, those are also perfectly fine and sufficient for the course. Okay. Keep in mind that this is a 6-week course and you don't have time to wait 2 to 3 weeks for a book to arrive, and that's why we strongly suggest an ebook for you to, at least have the material um, pretty much day one. If you look at the schedule, I believe your first exam is either towards the end of the second week, or even possibly the beginning of the third week. I forget off the top of my head, but it's right around the corner, unfortunately. The lab kits, these are going to be kits that will be sent to your home from Carolina Labs. The way this will work is once the bookstore is set up for this, you will have to contact them. They will give you an access code. You will go to Carolina's website and enter that access code with your home address. And it takes about 2 to 3 days for them to mail out the lab kit to you. So once we know that those codes are in the bookstore, please, please, please make sure that you order it from the bookstore within a day or so that we can start getting to doing the hands-on labs. Now, you will receive a box. It will have petri dishes in it. It will have bacteria in it. It will have various other things for microbiology, including a little microscope that you'll be using. So I ask that you do not open anything, that you do not really touch anything until you are instructed from me. Okay. The college is covering the costs of these kits for the summer, and like I said, they will be mailed to your home directly. Okay. And each student will only get one kit, as they are not that cheap. Okay, so chapter 1 is just an introduction. You should read over chapter 1. You should look over the PowerPoints. The way that you'll see each lecture has a PowerPoint associated with it. There will be a recorded lecture, which is what you are currently watching, hopefully. There is a review sheet for most chapters. I will warn you that chapter 2, chemistry of biology, is not something we typically cover face-to-face, even in our face-to-face lectures. It is more of a review for you to look over, as this was chapter 2 in either A&P 1 or general bio. Either one, which is a prereq for this course. It's just so that you can refresh your base knowledge of chemistry as it pertains to biology. The other thing that you'll notice is that there is a lecture assignment for each chapter, and then there is also a lecture quiz for each chapter. To make things easier on you, I have made the due dates for the lecture assignments and for the quizzes essentially Sunday nights. of each week module. Now, I do not recommend at all waiting until Saturday night to complete everything for that week. Although everything does have to be turned in by 11:59 Sunday night. I would stagger it throughout the week. Pace yourself. The content is a lot. We are covering a 15-week course within 6 weeks. So it is a bit intense, okay? And this is going to require you to pretty much be, interacting with the course on a daily basis for the next 6 weeks. In a normal summer semester, I would see you 4 days a week as it is, and 2 days in lab. So, it really is important that you are doing some of this, every day throughout the week, okay, with maybe taking a day or two off. Once a quiz closes, it will not be reopened for any reason, okay? If you miss the due date, you will get a 0. So it is very important that you do not miss any of these due dates. Same goes for the assignments. If an assignment or lab report is not submitted via Canvas by the due date, you will get a 0, okay? So it is very important that those are also submitted by the due dates. And again, for consistency purposes, I did make it Sunday at 11:59 of each week, okay? You'll see all the due dates on the syllabus within Canvas. If you click that little syllabus button, you'll see the due dates on the calendar of Canvas, okay? And on each of the lecture pages, you will see the due dates there. So there's no way you can tell me you didn't know that something was due. The due dates are listed, in 4 or 5 different places, okay? Please feel free to email me with any questions. I will try to get back to you as soon as I possibly can. I just want to warn you that I do have 100 students, so it might be a couple hours before I actually respond back to you, especially if I'm currently doing a recording or something. Okay, so microbiology is the study of microorganisms. Microorganisms are organisms that are too small to be seen with the unaided eye. So essentially what that means is you can't see it if you look at it, okay? You need to use an instrument, and the primary instrument that we do use is called the microscope. Something that you will become very familiar with, and one of the first labs that you're going to do this week is the microscope, and you're going to familiarize yourself with the microscope. Unfortunately, I don't think you're going to have your lab kits this week, so you might not actually get to use the microscope until week 2. But you will be able to familiarize yourself with the microscope, okay? But hopefully you've used the microscope in prior courses. Microbes include things such as bacteria, fungi, protists, algae, and viruses. And you might have seen this one a lot recent days, as COVID-19 is a virus. Unfortunately, I do see a lot of misinformation floating out there. Viruses are non-living entities, okay, so they cannot be killed. They can be destroyed, but they cannot be killed. And essentially what happens is viruses must have a host cell. And in this case, if we look at COVID, the host cell is going to be mucosal cells within the oral and nasal pharyngeal areas, where it will infect those areas, okay, and replicate. Bacteria are prokaryotic cells. We will have a chapter where we look exclusively on prokaryotic cells. Prokaryotic cells were cells that do not have a nucleus. They also do not have membrane-bound organelles. Fungi, these would be things such as mold and yeast, and they are eukaryotic cells. So right there is a distinct difference between prokaryotic cells and eukaryotic cells with bacteria and fungi respectively. And that's one reason why you can't use an antibiotic to treat, say, a yeast infection or an antifungal to treat a bacterial infection, okay? Protists, these tend to be parasites or parasitic. We're not going to get a whole lot into these guys as they are not that clinically relevant. Algae, we're just going to touch on probably in chapter 1 and briefly a little bit later on, but we're not going to get a whole lot into that. The third major category is viruses, and we will spend a whole chapter on viruses. And when we get to the actual diseases, you'll learn several viruses, including more about COVID-19. So microbes, very few are pathogenic. That is a term that means disease producing or has the ability to cause disease. Okay. If you look at the totality of microbes that are out there, very, very few are actually disease causing. In fact, some are actually beneficial to you, and most actually will have no real interaction with you one way or the other. They could, frankly, care less. Okay. Microbes do decompose organic waste. Bacteria and fungi break down any carbon-based material. Some of them are able to undergo photosynthesis, but a lot of the ones that we will actually talk about either will undergo aerobic respiration, a process we will go over again throughout this course, as well as fermentation, where we can produce things like ethanol and alcohol and various acidic products. Certain microbes have also been used in various manufacturing aspects. For example, I have insulin listed here. Insulin is actually made using Saccharomyces, which is a yeast. They took the human gene for insulin, they cloned it into these microbes, and they essentially turned them into little protein factories where these yeast cells actually produce and form insulin, which then can be purified and is what we now sell to diabetics so that they can inject insulin into them. Prior to this technology, people that took insulin shots were not actually taking human insulin. It was actually either bovine or swine. And like I said, this now is the human protein and various modifications depending on the different types of insulins that are out there. So knowledge of microorganisms allows humans to prevent food spoilage, helps to prevent disease, which this is big, especially if most of you are looking at going into nursing or allied health in some form or fashion. You will be coming in contact with various microbial diseases and will have to learn how to either prevent transmission of those diseases, but also prevent them from actually causing issue for you. Again, this is something that the general public is starting to really see a lot of today with COVID-19. Also understanding the cause and the transmission of the disease to prevent epidemics. Unfortunately, we are living through an epidemic, and we are still learning a lot about the actual transmission of the disease and actually how to prevent this. We know that you need to keep a distance. We know that you need to wear a face mask. We know that you need to wash your hands frequently. These are all good general practices when talking about any of these things. So microbes are easy and difficult to study, and you might find this course easy at times and very difficult at times. They reproduce very rapidly. They reproduce in very large populations. When you actually look at a petri dish with bacteria growing on it, it's not one or two bacteria that are growing on it. It's trillions of cells that can actually be grown. Unfortunately, they can't be seen directly. You will see lots of images of various microbes, but then when you guys get your lab kits, you're going to look at various very common bacterial strains underneath the microscope. And I can't wait to see all of your descriptions of those things once you get to look at them for yourselves. this is a new topic that has sort of been, and when I say new topic, I'm talking about the past decade. Microbiome. When I took microbiology in college, this wasn't even something that was really considered. But it's actually what we now refer to as all of the organisms that live on your surfaces or within you. Okay. And it's estimated that an adult human is composed of about 30 trillion body cells, and it can actually harbor another 40 trillion bacterial cells. And in fact, I've even seen some studies where the large intestines alone can actually harbor upwards of 100 trillion bacteria. So just think about that. You actually have more bacterial cells that are living within you than you actually have human cells. The microbiome is a group of microbes that actually live stably on and in the human body. They do help to maintain good health. We're learning more and more about these organisms that are part of our human body and the benefits that they provide for us. They actually are occupying a space that would be occupied by some type of microbe. And the saying that the devil you know is better than the one you don't know, having your microbiome in place and having a healthy microbiome in place can actually retard a lot of pathogenic microbes from taking up residence. It can also help to train the immune system to discriminate real threats, and it can help to prime the immune system as well. When I was in college, the microbiome was more commonly referred to as your normal microbiota or normal flora. And this is a collection of acquired microorganisms on or in a healthy human being. You start to acquire this as a newborn, especially as you're flying out through the birth canal. You are passing through the vaginal tract where you will pick up pretty much any organisms that your mom has on your way out. And that's really your initial colonization. But once you are actually out and you are, exposed to surfaces and people are handling you and you are crawling around and eating, your microbiome will develop. I always joke with my students in my face-to-face classes that the best thing you can do for a child is actually to throw them into daycare. They're going to get sick. But what will actually happen as a result of that is their immune systems will really develop nicely. They need to be exposed to these things, okay, for effective immunity. And that will tie into another topic that we'll get into, which is vaccinations. Vaccinations actually expose children at a very early age of a lot of these bad things so that they end up producing immunity. And in the event that they actually do see them in real life, they will have immunity against these things. And again, vaccines are something that, you might have heard a lot of in the news recently with the development of a COVID vaccine, but they've also been talking about how newborns and children need to continue getting their vaccine, even though we are sort of on a shutdown, because we don't want to run the risk of them actually contracting diseases like measles or rubella or things like that. So back in 2007, there was a Human Microbiome Project, and this was a multi-institution, meaning multi-hospital, multi-university institutions that asked a simple question. What is normal microbiota? What should we find on a normal individual? Okay. And unfortunately, there's no direct answer to that. It could be, lots of differences between males and females. There are differences between various age groups. So there's a lot of, variants that can play a role here. That project was slated for 10 years. And actually in 2016, it got re-upped under the National Microbiome Initiative through, I believe it's 2026. And we are actually learning a lot of neat information about these organisms that live on your surfaces. They are actually much more beneficial to you than many people realized. Okay, so some basics with microbiology. You're going to see lots of bacterial, fungi, or viral names, and they all follow the same nomenclature that we use in biology, where the entire name is italicized, as you can see here with E. coli or Staphylococcus aureus. If it's abbreviated, the genus name, which is the first part of the name here, is always capitalized, and it's just the first letter if it's abbreviated. The species name is always spelled out. So here, instead of saying Staphylococcus aureus, Staphylococcus is the genus, aureus is the species. Here you can say S. aureus, okay, where you're just listing the species name with the abbreviation for the genus. So the different types of microorganisms are bacteria, archaea, fungi, protists, algae, viruses, and multicellular animal parasites. There are a few on this list I didn't mention on the initial slide, but we're going to go through each one. So bacteria are prokaryotic cells, and I'm not going to go into too much detail with each one of these right now because as if you look, Chapter 4 gets into prokaryotic and eukaryotic profiles, and Chapter 13 gets into viral profiles. So bacteria are prokaryotic cells. They are going to exist as single-celled organisms. They do have a cell wall. The cell wall is very important in that it protects against osmotic stress. The cell wall is also a unique structure when we look at human cells. Human cells are animal cells, okay? Animal cells lack cell walls. And because bacteria have cell walls, and as you're going to see in a second, fungi have cell walls, we can actually target the cell wall with drugs and disrupt the cell wall. And by disrupting the cell wall, that can actually lead to death of the cell. The idea here is to target something that would not affect human cells. And because humans do not have a cell wall, if we target the cell wall, we're not affecting our cells. Bacteria will still have a plasma membrane. All living cells will have that structure, as do human cells. Hopefully you are aware that human cells have a plasma membrane, cytoplasmic membrane, whatever you want to call it, cell membrane. It's the same thing, okay? So if we were to target the plasma membrane of bacteria, because it's composed of the same thing that human plasma membranes are composed of, if you use a drug to destroy that structure, it most likely will also destroy your cells. Okay, so not a good drug target. Bacteria will divide by a process called binary fission, which is a very simple process. We'll get into that in Chapter 4. But binary fission, essentially, the cell grows and splits into two. You don't have mitosis, you don't have meiosis, you don't have any of those complicated processes that you typically see. Bacteria may be able to move or quote-unquote swim by structures called flagella. And here's a picture of some bacteria. Archaea are also prokaryotes. They lack cell walls. They live in very extreme environments, such as areas where there's a lot of salt, or areas where that's halophiles, there's a lot of salt, or areas like hot springs where there's a lot of high temperatures, that would be thermophiles. We really don't get into these beyond this slide here, so you will probably never see these things again. Just know that generally what they are, okay? Fungi, these are eukaryotic cells, okay? And with the fungi, they are very closely related to your cells. Because they are eukaryotes, we have a lot of similarity between the two cell types, which unfortunately can, at times, make it difficult to find drug targets. They also have cell walls. Now remember, animal cells do not. And if you look, cell walls are chitin, which is that word C-H-I-T-I-N right here, okay? In prokaryotes or bacteria, the cell wall is peptidoglycan. And If you break that word down, peptidome means peptides, which is proteins. Glycan means sugars. So this is made up of proteins and sugars. Chitin, which is a cell wall found in fungi, is made up of polysaccharides. Chitin is very similar to cellulose, okay, which you might be familiar with as being the cell wall component that we typically find in plants. Fungi will include your molds and mushrooms, which tend to be multicellular organisms, and as well as yeast, which typically are unicellular organisms. And here we can see this actually mold, where you can see some spores and the hyphae formations. Protists are also eukaryotes. They will absorb or ingest organic chemicals. They may be motile via pseudopods, cilia, or flagella. They are free-living or parasitic, and some are photosynthetic as well. They may reproduce sexually or asexually. We're not going to get a whole lot into these guys. Again, just know what they are. Algae, these are eukaryotes as well. Algae mimic plants. If you notice what their cell walls are composed of, it is cellulose. Again, cell walls protect against osmotic stress. I think that's the third time I've mentioned that. And animal cells do not have cell walls, which include human cells. Okay. Algae will undergo photosynthesis, which again is a process of energy production that plants undergo. And we can see both sexual and asexual reproduction for algae. And here's, if you look at a lot of algae underneath the microscope, it's actually very pretty on what you might actually see. Then we get to viruses. Viruses are acellular, which means they are not made up of cells. They are consisting of either DNA or RNA, but they will never consist of both components. It's either one or the other. We call that the core of the virus. The core of the virus will then be surrounded by a protein coat, which is called a capsid, and the coat may be enclosed in a lipid envelope. And if it does have that lipid envelope, it's referred to as an enveloped virus. If it does not have that lipid envelope, it's referred to as a naked virus. And again, we'll get more into that when we get to chapter 13. All viruses must have a living host cell, okay? And despite what Lysol says, or our illustrious president, Lysol does not kill viruses, okay? Lysol will destroy viruses. That's why when you spray Lysol on a surface, what it does is it breaks down the protein and nucleic acids. What they're telling you to do when you wash your hands for 20 seconds with soap, it doesn't have to be antibacterial, you just need soap, because the soap will disrupt the protein composition of the viruses and it will break it down. Okay, the 20 seconds is making sure that you are giving it enough time to dissolve those components. Once a virus breaks down, it is ineffective. It needs to be fully, it has to maintain its structure for it to be effective. Okay. And viruses must have host cells. If you no longer have host cells for the virus to replicate in, what happens is the virus just sort of goes away, which is the whole theory behind these stay-at-home orders. If we can reduce the amount of people being exposed to these viruses, especially COVID-19, what you do is you tend to flatten the infection curve, which I'm sure you guys have heard of in the news. Okay, parasites. We're only going to talk about this when we get to diseases of the GI tract. Parasites are going to be things like your worms, okay? And I will leave you with this little fact for now, that 1/3 of the world's population actually has a worm that lives inside of their intestinal tract. Just leave you that with that little fun fact there. So classification of microorganisms, they are broken down into either bacteria, archaea, or your eukarya. Your eukarya will include your protists, fungi, your plants, which will include algae, and any animal cells, which also include humans, okay? These are the three main domains that exist, okay? Okay, so the next couple slides just go over some important people. I don't want you guys to focus too much on this. I'm not going to ask you any specific dates on a test or anything, but you should know some important people that were, integral for microbiology. And the first one is Leeuwenhoek. Anton van Leeuwenhoek was the first guy to actually develop a microscope. And by developing a microscope, he allowed us to be able to see these things that we are not able to see. Unfortunately, or fortunately, he did such a great job that the microscope that we typically use in a lab, and the one we would have used in our face-to-face lab, is not much different than the one that he developed. We essentially use a standard light microscope, which is called a compound microscope. It's called a compound microscope because it actually has two different lenses. You have the two lenses that you're looking through, and then you have the objective lenses, which you can change. And your magnification is multiplication of those two lenses together. So if you were looking at a 40x objective and your ocular lens, which is the ones you're looking through, is 10x, 40 times 10 is 400. And your total magnification would be 400 times. Some other important people here, Louis Pasteur, you will hear of him in a little, as we go through this course, for two main things. Pasteur came up with a process called pasteurization, which is something that we use for a lot of our dairy products. It essentially reduces the number of microbes that are found, say, in milk, and it extends the shelf life of milk. The other thing that Pasteur is important for is that he actually didn't discover, well, maybe he did discover. It's not like he created it, though. He characterized fermentation, which is a process in which microbes can take sugars and, say, convert them into alcohol. Okay. Joseph Lister, he was a surgeon, and he actually was he's dubbed as the father of disinfectants. And the reason why he's listed as the father of disinfectants is because when he did surgery, he realized that, say 50% of his patients would get an infection and die. And he realized that maybe by cutting them open, they're getting infected with these microbes. So he came up with, we need to disinfect the instruments, we need to disinfect the surfaces, and so on and so forth. The third main individual is Robert Koch, and Robert Koch established experimental steps for directly linking specific microbes to specific diseases. So in essence, he's the guy that said, these things that we cannot see actually can make you sick. Okay. And here you can see various names. You'll actually might see some of these names as you go through this course. For example, Graham, he came up with the Graham staining procedure. Petri developed a Petri dish, so on and so forth. Another important person would be Alexander Fleming. Alexander Fleming was the first guy in 1928 to actually discover the first antibiotic. And this was breaking. I mean, this was a breakthrough, because if you ended up with a bloodstream infection or an infection that got into your blood, respiratory tract, it could kill you. And they had no way of stopping that. And then came along antibiotics, which would be administered and would essentially allow the individual to overcome the infection. Penicillin was initially discovered by accident. He was a bacteriologist, and he would culture bacteria. And one day on one of his petri dishes, he noticed this contaminant down here. That's this big white blob down here. And unfortunately, with this contaminant, this is a mold. And this mold actually, if you look at the bacteria that surround it, they're not really growing compared to up here. Now, he found that to be interesting. And he's like, why aren't these bacteria growing when they're close to this guy? And we now know that this guy secretes a protein that inhibits the growth of these bacteria. And we now know that protein to be penicillin. Okay, unfortunately, with the onset of antibiotics and the use of antibiotics and the broad use of antibiotics, especially in the 70s, 80s, and 90s, where they were sort of administered for every little thing, we started to see resistance and drug resistance. And this will be a topic that we discuss throughout this course, is the problems with antimicrobial chemicals. And the main problem is the ability of drug resistance to develop. One thing I just want to leave you with today on this is that the resistance is with the organism. It's not the antibiotic. It's the organism that is not responding to that chemical anymore. Okay, and it's also not you. It is the bacteria is what is resistant to the antibiotics. All right, the various divisions of microbiology. You have bacteriology, which is the study of bacteria, mycology, which is the study of fungi, parasitology, which is the study of protist and parasitic worms. Immunology looks at the immune response in the immune system. So this is, as a human, your response. Ernie. And virology is the study of viruses, okay, which is actually a fairly new field comparative to the other three. And the reason for that is we were not able to see viruses under a microscope because they are very small. As you'll see when we get to chapter 13, a typical light microscope, even if we were in a face-to-face lab, you would not be looking at viruses. You need something called an electron microscope to be able to see these. And we, until that was actually developed, we never really were able to even see these things. Okay, we will get into, with this course, I believe it's chapter 8, looking at the genetics of microbial genetics, which is, how do their genes differ from ours? There's really not a lot of difference. There's actually some conserved pathways that we have that also E. coli will have. But we'll get into that. This is more of a chapter 8 thing, okay? And then, we'll look at other functions of microbes, for example, things called bioremediation. Bioremediation is using microbes to clean up pollutants. Sewage treatment, this is a bioremediation as well. Sewage is 99% water, and then you also have things like bacteria, protists, fungi that are present there, and other organic material in sewage. And believe it or not, these microbes actually help to break down a lot of that organic material and help to clean the water, before it gets to, say, a faucet where you might be drinking it. So these things actually do play a role in recycling of water. Obviously, there's a lot of more purification steps involved there along the way. Biotechnology, this is chapter 9 in the book. I don't get into this a lot, but this is essentially, for example, that example I gave you where we use yeast to actually produce insulin. This is where we took the human gene for insulin, cloned it into yeast, and able to produce that protein. Okay. The normal microbiota, we talked a little bit about this. This will be a topic that we talk about throughout the course as well. You will hear microbiome as a consistent theme throughout the entire course. Okay. Biofilms, sorry, slides got a little away from me. These are communities of microorganisms. They tend to attach to surfaces. We will get into some clinical areas where biofilms will also play an important role. But an example of a biofilm is when you woke up this morning, that nice slimy layer on the roof of your mouth or on your teeth, that's a biofilm. Okay. And depending on the surface, you want to make sure that gets disrupted because it could become more invasive. All right, emerging and new infectious diseases. Here's a list of all diseases, and when we look at what actually affects the U.S. versus what affects worldwide, the list may not be the same. The U.S. is a developed country. There are a lot of developing countries and second world order countries which don't have the healthcare systems that we have, although we can see that the U.S. is not infallible as well. It's evident by COVID. There are always new and emerging infectious diseases. Unfortunately, we are currently living through this time where we are living through an emerging infectious disease, which is COVID-19. So we will talk about that topic as well. So there are always new things that pop up. I always say to my class, it might be interesting to see what pops up while you're taking this course. Unfortunately, a current thing with COVID-19 that you are currently living through is something that has forced our class being from face to face to an online course, and this is something that we are currently living through. Okay, so that's the end of chapter 1. I hope you enjoyed that lecture, and like I said, you want to keep up with these lectures. Okay, you want to make sure that you are looking at these things. Okay, and don't wait till Saturday night to complete all of these things. You want to make sure that you're breaking up your time and using it wisely throughout the week. Module 1 has 4 chapters in it. Granted, one of those chapters is a review chapter, but again, we are condensing 15 weeks worth of material down to 6 weeks. Okay. All right. Have a good one.